I'm Mr. Grobler, and I'm here to ask you a question. If you had to select a topic in life sciences that you would say is the foundation for everything in life sciences, the basis to build on for knowledge, what topic would that be? Let me show you. It's all about cells and cell division. We'll be looking today at the basic unit of life. Cell structure we'll look at. We will look at the government of the cell, the nucleus, and then the role of the nucleus when it comes to cell division, which we call mitosis, and that leads to growth. And then we will have a look at what happens if mitosis is out of control. So before we start, I want you to look out for a few key points in this discussion, some terminology that are important. Let's look at these words. Can you really explain what is a cell? Think about it. A big part of the cell is the cytoplasm. And then floating in the cytoplasm are the organelles. And then we will look at a very important terminology in life sciences, which is osmosis, and that has to do with the movement of water in the cell. So I asked the question, do you know what the cell is? Let me show you. The cell is the smallest living unit of any living organism. Whether it's a plant, an animal, or a human being, look at these building blocks of life. Here's a cell. Here's another one. Here's another cell. So each cell is like a brick building a wall. And that's really what you and I look like inside under a microscope. Now, looking at the next definition of what are these organelles? Organelles are these specialized structures. If this is a cell, the little compartments that we have inside a cell, these structures are called organelles. Now, if you would remove, let's say, a muscle, or let's say muscle tissue, and you would go smaller and smaller until you would find the muscle fibers or cells, could be skin cells, it could be the cells inside your mouth. If you look under a microscope and you would examine all the structures inside a cell, these are so small, and yet it's so complex, it can be compared to a whole city. Let me show you what I mean by that. If you look at the cell here, look for instance at part number 1. That part can be compared to a storeroom. That is for storage. Look at part number 3. That could be compared to the municipality of a city or the governance in a city. Look at part number 4. That could be compared to a factory. And so we look at number 5. Number 5 can be compared to roads. A road system in a city. Number 6 is the army. Number 7 is a toolbox. And look at number 11. That is the power station, providing the energy for the cell. Do you realize how complex a cell is? And it's fascinating to study it. Now, what you will be required to do throughout life sciences studies is to be able to label diagrams and know the functions of the different organelles floating in the cytoplasm. Now, the cytoplasm is often a liquid within the cell membrane, and 70% of it is water, actually, of the cytoplasm. Sometimes it becomes more like a jelly, but the little structures called organelles are floating in that cytoplasm. Now, we're now going to look at the labels and the names of these different organelles and their functions. So let's have a look. Here the question comes in an assessment. Name the organelles, and the numbers are given there, number 1, number 5, and so on. So while we're discussing these different numbers, I want you to also listen to the functions. So I will now make a list of these labels, especially as this question paper is asking it. But while I'm speaking and telling you about the names of the organelles, I will be also summarizing the functions. And just after that, I'll be giving you a short test to see if you listen carefully to pick up the functions of each of these organelles. So here are the organelles. Number 1, please look at number 1 in the diagram. Can you see number 1 is the storeroom? Storing what? Water. Can you see the network number 5, which I called like a roadway? Transporting. And this is what the ER is all about. It's all about transport. Now, if you look at that label number 5, can you see I use an abbreviation called the ER? Now, ER, you can use it like that in the exams, but it stands for endoplasmic reticulum. It's all about transport all the way in from the outside cell membrane towards the nucleus. Look at number 7. Now, number 7 we'll deal with more when we talk about cell division. It's all about playing a massive role in the process that we will be discussing just now, mitosis. Now, look at the small structures that you see around about number 8. These structures are ribosomes, and this is the place where proteins are produced. We talk about protein synthesis. And then number 11 is the power station. It is for the production of energy. So let's now move to the next slide to now look at the functions and to see if you can actually remember the functions of these different parts. Here's the question. Typically in the exam, provide one function. Now the function is the job. Some people are not too sure what the word function means. The job each of these little organelles are doing inside, like organelle number 1, 5, 7, and so on. So let's hear from you. Can you please shout out where you're sitting and viewing this lesson? When I say a number, please shout out the function there. Whether you're sitting alone or with a group of people, just say it loud. What is the function of number 1? I think you're right. What is the function of number 5? Can you remember what I said just now about number 7? It plays a role in which process? And then number 8, there's a production of something happening at number 8. And what about number 11? What is the function of number 11? How did you do? Did you get them right in that quick fire round? Let's check out the answers. Number 1, the vacuole stores water. Number 2, remember the ER is for transport into the cell. That is number 5, the label number 5. Number 7, remember that structure there, the centriole, is all about cell division. They play a massive role in cell division, which we call mitosis. And then number 8, the production of what? Proteins. The word synthesis means production. And then we said number 11 is the what? Number 11 is one mitochondrion. And the mitochondrion is all about producing energy. And that is during a process that we refer to as cellular respiration. So now that you know some of the basic organelles and you know their functions, let's look at a few other typical assessment questions that you can get in projects and practicals, exams and tests. Here comes a typical question. Tabulate 3 differences between animal cells and plant cells for 7 marks. Why is it 7 marks? Because the moment we say tabulate, you must draw a table. Give it a heading. And write here clearly, this is, let's say, a plant cell on the one side, and then you compare it to animal cell. And whatever you write on the one side must be to the same topic as to the other side. You must compare the same topic here to the other. Now, what I will be doing now, I will show you the animal cell that we've been discussing so far, and then I'll show you a picture of a plant cell, and then you spot the differences. And I think you're going to get 7 out of 7. There's the animal cell that we've become familiar with so far. Now see how it differs from the plant cell that we have here. First of all, do you not see a cell wall here? There's a cell wall on the outside which the animal cell did not have. That's already a difference. Now look at the vacuole here. The vacuole we saw in the animal cell was small. The vacuole we see here is big. So that is another difference. There is a large vacuole or vacuoles in plant cells. And then we can look at these green structures here. Do you remember what they called? The label tells you these green structures, these organelles are chloroplasts. So there are also other plastids. There's chloroplasts, but there's also some other plastids, specifically in plant cells. Animal cells don't have them. So now we're ready to draw our table. And let's see what it will look like. And this is really where you can do very well in the assessments. Look here. We provide a heading, difference between plant cell and animal cell. So we always provide any table with a heading. And then we write on the one side plant cell in comparison to animal cell. Difference, we said plant cell has a cell wall, while the animal cell has no cell wall. I know that you know this. Plant cell has a large vacuole, while animal cells is small. The plastids are here in the plant cell, and there are no plastids. I think so far you've done well. Remember, I tested you on the knowledge of, when I gave you the labels, you had to give the functions. I think you probably did well there. And when it came to the table, you scored more marks there. Now, when we study life sciences, it's not only about scoring the marks, but I know it's a bonus when you can do well. Let's look at some more exam questions. Describe the movement of water, because we said water makes 70% of the cell is water. Now, describe what happens with water if you put this cell in a strong salt solution. So where's the salt solution? It is this part which I've indicated here. Now you go and you place the cell in the salt solution. Now what the salt solution does, it causes a low water concentration. Now on the inside, the cell has more water. So where will the water go? It always goes in this direction, from high concentration to a low concentration of water. So inside the cell, it is normally higher than outside, especially if you put it in a salt solution. Now sugar does the same. Salt or sugar solution causes water to leave the cell from inside to out, because the salt or the sugar causes a low water concentration or low water potential. So let's just describe this in a paragraph type of question. Water moves by means of which process? Osmosis. Through a selectively permeable cell membrane, which means it chooses what can go through. Now the salt solution causes a what? A low water concentration outside. Now you've created from high to low, and the moment you talk about high and you talk about low, you say there is a gradient. And the result is water moves out of the cell. Let's just recap what we've done so far. Can you now tell what is a cell? I'm sure you can, the smallest living unit. The cytoplasm is the part inside, mainly consisting out of water. The organelles are those small structures inside the cell, and we said that osmosis is the movement of water. I think you have a good understanding so far. Now we're ready to move into the government, the whole control center of the cell. I said it's like in a city, the municipality. We'll be looking at what's happening inside the government, inside the files especially there. So when you come back after the break, we'll be focusing on the nucleus. So welcome back. Let's continue with our discussion. And we said when you come back, we'll be looking into the government, the secret files, and we will see what they are called in biological terms. Let's have a look. So far, we've looked at the different organelles inside the cell. Now we really want to focus on the government, because the government controls processes like growth, which we call mitosis. And we want to focus on that now. So there's a few key terminologies that we need to understand. And when I show you these words, I want you to understand how they link to one another. Don't examine them separately. These structures are linked to one another. And you need to understand how they are related. Let's have a look. First of all, we now know the government of the cell is the nucleus. Don't worry about this word now. We'll come back to that because that's different from the nucleus. But how do these words relate to one another? nucleus with regards to chromosomes, where does the DNA fit in, and what are the genes we're talking about. We will talk about cell division a bit later on, but these that I've circled here, I want you to understand how they relate to one another. So let's go into some diagrams, and when you see the basic structure of the cell now, you will see where the nucleus is and where these other structures are found. The structures below carry genetic information, so it's all about information. It's like we can compare it to files. That is the comparison we can make, which controls what? All the processes inside the plant or the animal or the human being. Now label the structures. Can you see what number 1 is? Is a cell membrane. So inside it represents the cell. Then obviously number 3 is the nucleus. But I want you to focus on number 4. I've taken one of these, number 4. Now number 4 you see occur in a pair. So number 4 here I've drawn 1 chromosome. And we want to look at the labels of chromosomes, but I want you to understand where the chromosome fits into the nucleus. Can you spot the nucleus? Yes, it's label number 3 with a nuclear membrane, and the nuclear plasm is number 5 inside it. But before we carry on, let's just put these labels in a logical order. Let's make a list of these labels. So there you see the cell membrane on the outside. Everything on the inside of the cell membrane is, of course, the cytoplasm there. And then inside the cytoplasm, which you can see there is number 2, is the nucleus with its membrane on the outside. And what you see is that important label number 4, the chromosomes that you can see there. Of course, in comparison to the cytoplasm, what's inside the nucleus is the nucleoplasm. But what we're now going to do, we're going to look at one single chromosome. I must say the chromosomes come in like shoes in pairs. But we will be drawing the sketch of one chromosome now, and see if you can label and understand how the chromosome fits into the nucleus and then break it down into its smaller parts. So here, look at the labels here. Can you see what number 6 is called there? If you look at the sketch number 6 in the diagram there, these are chromatids. So the centromere is number 7 that you can see there, binding the chromatids together. Now somebody will be tempted, if you look at number 6, somebody will be tempted to say there are 4, but no, there are 2 chromatids. Here I've drawn one, and here's the other one. Now the chromatids consist out of what? As they're being bound together by the centromere. They consist out of each one, as look at number 8, a single DNA molecule that's spiraled around here. This is a single long chain molecule, a chemical called DNA. Now, the DNA contains the codes of life. It carries the information, as we said, in code form about you. What makes up you as a person? And this was written in your genes in code form on the DNA before you and I were even born. So what do we mean? It's all about, if you would send a text message, you have 26 letters of the alphabet that are available to you to use in a text message. But when it comes to the way the codes have been written of life, for instructions how tall you must be one day, what will be the shape of your little toe, the size of your ears and everything like that, there are 1 million genes which carry the codes not in 26 letters of the alphabet, but 4 letters. Let's look at these 4 letters. I'm going to abbreviate them. A stands for adenine, but you don't need to know that for now. There's T, there is G, and there is C. Now the order in which these letters appear here on the chromatid determines a certain code for, let's say, eye color. Now reading it down like that is what we call a gene. These are the secret codes of life. So do you now see how the chromatids that you can see here, number 6, they contain the DNA molecule. They're bound together by number 7. Look at number 7 label is the centromere. But the DNA contains the genes, which are the codes of life found in these 4 letters. I think you have a good understanding how the bigger picture is getting smaller and how they all fit together. Let's look at some more questions with regards to the nucleus. How many chromosomes are there in the normal human body cell? Here comes the answer. There are 46, but we like to write them in pairs because they are like shoes. Chromosomes come in pairs. Many learners make a mistake. They forget, want to write 23, but it's not 23, it's 23 pairs, which means 46. Now, there are more typical questions that us as examiners like to ask, and let me show you. Name 2 functions of the DNA that's found on the chromosomes. Why is the DNA there? The DNA controls everything. It controls the production in that cell, especially of important proteins. The DNA is also responsible for the transmission of hereditary characteristics. Now that means that you are born with. When we talk about heredity, it means what you are born with. It's the DNA that carries the information from parent to whom? To offspring. Do you now see why we love our parents so much? Why we need to respect them? Because what's inside us, the codes of life, which we call the genes, which are written in code form on the DNA with those 4 letters A, D, G, and C. So it's A, T, G, and C. If you think in terms of that, you understand where you come from, and then you respect your parents. So keep on doing that. Keep on thinking where do you come from, and then show that love to your parents. Let's move on to a summary of this. Answer the following questions now on what the nucleus does when it comes to mitosis. So what is mitosis is the first question. And the question is, why is mitosis so important? The biological importance or the significance is simply, why is mitosis so important? And can you say what it is? Mitosis is a process whereby one cell divides to form 2 cells. But the important thing about this cell division is this cell here must be identical to that one and must be identical in that one. In what concept? In about the DNA that's found there. The DNA that's found in all of them must be identical. So there must be a copying process, like a photostat process here, before cell division takes place. But here we see, what is mitosis? It is the dividing of one cell to ultimately form 2 cells. Now the original cell is called a parent cell. Some people call it a mother cell, parent cell. And what is formed here in the end are 2 what? We always talk like this. Doesn't matter if it's male or female. 2 daughter cells. So why is mitosis important? It's important for growth, because if you're going to have one cell and you can multiply it to 2, and the 2 can become 4, now you're building bricks, putting bricks together for growth. Mitosis is also important to repair damaged tissues. And that is a wonderful ability your body and mine has. You scratch yourself and then new cells form to repair itself. Let's now summarize everything. Describe what happens from the moment a new cell is formed until it's ready to divide again. So a new cell is forming. Now, what is going to happen when it divides again and again to form more and more cells? Now, each of these can divide. The 4 can become 8 and the 8, 16 and so on. Let's look at the pictures here. Once a cell is formed, and this actually brings everything together, it grows to its normal size, but then, as soon as it's now preparing to divide again, it must first go into an in-between period called the interphase. Now, what happens during the interphase? This is when the DNA copies itself. DNA replication is to make copies of the DNA so that when new cells are formed, that the new cells have the identical DNA than the original mother cell. Copies have to be formed, and that's why all the cells carry the genetic information similar to their parent cells. So this ensures that the chromosomes of the daughter cells after mitosis are the exact what? Exact copies of the mother cell. So can you remember? We've been talking about the nucleus, and then inside the nucleus, those structures that look like this are found. There's one chromatid, there's another one bound together with a centromere. That is a chromosome. Remember, they come together, they occur in pairs, and on there is spiraled the DNA molecule, which carries the genes written in code form. All about you. And we spoke about why mitosis is so important. But one thing I did not highlight was what is the difference between nucleus and nucleolus. Now the nucleolus, let's say this is a nucleus. Now you have the nucleoplasm here. Then also in here, you have a little structure called the nucleolus. So this label here is...
Yeah.
Yes! So crazy. You know, I tell myself before the time.
Because, what is it?
Yeah.
Old guys on the just...
There's so many things that distract you, you know.
Yeah.
Right, so...
Let's just start with this.
So I will just introduce it. Or should I just go to the slide itself? I think I just go to the slide itself.
In the beginning, what did you do? Did you introduce this slide?
Yeah, but you still have that introduction there. So we'll just go to it.
Okay, no problem.
So,
3, should I start with?
yeah.
Okay, 3, 2, 1.
So now we want to see how these concepts interrelate. If you think in terms of the nucleus that we saw, where really are the chromosomes? Remember, a chromosome looks like this, and the chromosomes occur in pairs, which we call homologous pairs. Then the DNA we saw is spiraled around here, a chemical molecule, which carries the genes in the form of codes, these 4 codes only. And the order of this is extremely important to determine ultimately what you're going to look like. We also spoke about the importance of cell division. Why really should mitosis take place? But there's one thing I did not mention here, and this is this word nucleolus. How does it compare to nucleus? So let's just look at that. If this is a cell, one single cell, you have the nuclear membrane, which is, by the way, actually a double membrane. So there I've drawn the nucleus now. And inside the nucleus is a small nuclear body containing RNA. We've learned about DNA. But this nuclear body inside the nucleus, so that part is the nucleus, the government we spoke about. But inside the nucleus is a nuclear body containing RNA. Now that nuclear body has a name, and that's called the nucleolus. I think that clarifies then the last concept among these. So when we get back after the break, we will be looking at the process of mitosis itself. See you just now. So we've been referring to mitosis. If somebody asks you, what did we discuss today? And we say cells and mitosis. Do you really know what mitosis is? It's cell division. Now that is what we will be looking at now. How does the process actually happen for you to grow? Remember when you were born, you were much smaller than now. How did the cells multiply up to the point where you now have in your body millions upon millions of cells? Let's look at that process, but before we go into that, we just want to recap again what we've done so far. We've been looking at cell structure with all the organelles. We spoke about the government of the cell with all the files in there, secret files containing the information called genes. We referred to the importance of meiosis, but now we want to look at the process of meiosis. How really does it happen, And what happens if something is out of the ordinary when it comes to cell division? Now, there's a few words that you need to understand when we talk about mitosis. Let's look at these words before we go into the process itself. You need to know the 4 phases of mitosis. We're starting off with the prophase. Then we will be discussing what happens during the metaphase. Then there's the anaphase, and then we come to the end, the telophase. You need to know the names of these 4, and you need to know what happens in each of them. What is the role of the centrioles? Can you still remember what the centrioles are? These are tools for cell division. Then cytokinesis. It tells you it's something to do with the cytoplasm. And then when things can go slightly wrong, but it can be corrected through medical treatment, is uncontrolled mitosis that can be corrected. So we now want to look at what happens really when one cell becomes 2. It runs through those 4 steps: prophase, metaphase, anaphase, and telophase. Let's have a look. The first one says, draw a label diagram, because in life sciences we do expect you to draw diagrams of which phase. We're starting with the very first phase, the prophase of mitosis. So how would one draw it? The first phase is the prophase, but now you want to describe by means of a diagram, you want to show in a sketch, by using labels, what really is happening here. Now, the first thing that we see is that the chromosomes become visible. We see the nuclear membrane starts to break up, as you can see with my dotted line here. Then spindle fibers start to form between these structures which we said before. Can you remember what these structures are called here? There's one here and there's one on the other side as well. Do you remember they're called what? Centrioles. Yes, one centriole, and then there's the other one that side as well. The 2 centrioles. So the chromosomes are now visible, and the centrioles will be moving one to the one side of the cell and the one to the other side. We refer to the 2 poles of the cell. That's where the centrioles are moving to. So what is the main event of the prophase? It's all about the chromosomes becoming clearly visible, and then the nuclear membrane starting to break up. So what is the next phase after the prophase? Did we not say metaphase? Let's see what happens during the metaphase. Here comes a question in the typical exam paper. Describe now in words what happens during the metaphase which follows after the prophase. Remember, the prophase was the first phase of mitosis. Now we're busy with the metaphase, which is the second phase. Clearly, you can see here, the one centriole is now, you can see, positioned in the one pole of the cell. That means the one side, and the other centriole is on the other side. The 2 chromosomes find themselves in the middle of the cell. We talk of the equator just like planet Earth. So look at the chromosomes here. Here's one pair, but remember in humans we have how many pairs again? 23 pairs of chromosomes arranged in the middle here, and they all attach themselves on these fibers called spindle fibers. So what's the main event of the metaphase? The chromosomes arrange themselves on the equator of the cell. They attach to the spindle fibers, and that is basically the end of it. So if we can describe it in a paragraph form as the question paper asked us to do, how would one put the thoughts converting it to ink on paper? Let me show you. The centrioles move to the opposite poles, and please, some learners are so silly to talk about the north pole of the cell and the south pole of the cell. No, that's planet Earth. That is totally wrong. They move to the opposite poles of the cells, and these centrioles, as they move from the one side to the other side, form what we call spindle fibers, very much like pieces of string between the two of them. And the chromosomes now arrange themselves in the middle line here. That's where the chromosomes are going to lie. And they attach themselves like somebody holding onto a piece of string or rope. So remember we said we started off with the prophase. We've now discussed what follows after the prophase, the metaphase. But what happens during the anaphase is quite fascinating. Let's see. Now the diagram below represents the phase of mitosis that follows after the metaphase, and that is the, step number 3 of mitosis is the anaphase. Now you can clearly see when you look here what is happening during the anaphase. If you remember the one chromosome, let's just look at one chromosome with its chromatids. There it is. Now what's happening during the anaphase is that the centromere here in the middle splits. And the one chromatid moves to the one pole of the cell by means of these little pieces of string called the spindle fiber. And the other chromatid, because the centromere split in the middle, moves to the other pole. And here you can see it clearly. Can you see this one is moving in this direction, this chromatid? And this one is moving in this way. It has split here in the middle at the centromere. The other chromosomes will do the same. And in the human being, all 46 of them will do that. They will split here in the middle at the centromere and separate like that. That is what happens during the anaphase. So do you remember, we start off with the prophase, then comes the metaphase. Now we've done the anaphase, and the last phase I mentioned in the beginning is the telophase. Let's see what happens in the telophase. So we said the anaphase was that last phase we saw. Now what is the function of the spindle fibers during the anaphase before we move over to that very last phase called the telophase? The function of the anaphase of the spindle fibers is to pull like pieces of string, to pull this in the direction of the two poles. So the spindle fibers shorten, and in that way they pull the chromatids to the opposite poles, ultimately moving towards that last phase called the telophase. So the anaphase is building up towards the last phase, the telophase. So how does the anaphase now contribute to the fact that the daughter cells that's going to be found in the telophase have the same number of chromosomes as the parent cell? So you can already see this whole division here that took place in splitting the chromosome at the centromere and there as well. And then there was one, so one chromatid moves there and one chromatid moves there. It was one chromosome. But so the numbers will stay the same, that will end up at the poles this side to that side. We started off with 2 chromosomes, and now it's chromatids that are also 2 in number that side, and the 2 chromatids that you can see here that will be ending up on this side of this pole. So here comes the answer with regards to the anaphase. Each chromosome splits at the centromere. The chromatids move each one to the opposite poles. In humans, the cell goes into mitosis with how many chromosomes? 46. There are now 46 chromosomes that have each split up to form the same number, 46 chromatids in the daughter cells. Now the DNA copies itself. If you have a shortage of copies at school, you must go to the photostat machine and copy. This is what DNA replication is all about during the interphase. So what's going to happen later on is those chromatids that would look like that, when they get to this interphase and the DNA copies itself, it makes a copy to add the other one. Now that is a copy. This chromatid, this side, is a copy of the original DNA that was spiraled here around this chromatid. It's simply copied during the interphase so that you can have complete chromosomes. In that way, the chromosome number stays the same. So we move on to the telophase. The telophase is the end of the whole process. And this diagram illustrates, because the question says, once again in life sciences, we are required to draw sketches. So if you think of how this will now divide, does it not make sense that the cytoplasm must divide in here? And that process we spoke about is that word we call cytokinesis. Cytokinesis is the process when the cytoplasm splits in here into two. And ultimately we're going to have one cell forming here with its own nucleus and another cell forming there. Now we have two cells, each with its own nucleus, and still the correct chromosome number, because we started off with 2 chromosomes and we're ending also with 2 chromosomes in here. These are chromatids that's going to copy themselves, and you're going to end off with 2 chromosomes. So, in summarizing the whole process, let's see how it runs from beginning to end. When we talk about this process, remember we ran through the 4 phases of mitosis: prophase, metaphase, anaphase. Can you remember the last one? Telophase. Let's start from the beginning and run through it again. Is the prophase. Then it goes on into the metaphase. But what is the interphase? The interphase is like a break. Just like you have a break at school before you go to the period after break. There's a break, an in-between period. And what happens during this interphase? During the interphase, DNA replication happens. And we saw now why the DNA copies itself. This happens in the interphase. But when we talk about mitosis itself, mitosis starts here with the prophase. And you remember what happens during the prophase. Then follows the metaphase. And then as we continue, the metaphase then is where these centromeres split. Where the centromeres split is the anaphase. Now they start to separate from one another, these chromatids, and they move to the opposite poles. During the telophase, they have grouped themselves now at opposite poles, a new nucleus form, and now cytokinesis can happen, and you have 2 new cells. There's one, and in fact in the end, it's 2 new cells. How many of them? 2, because there's one and here is the other one. 2 new cells have been formed. So in looking at the telophase, you can now see what I'm talking about. Here's one cell at the end, and what is important about these 2 cells, these are identical to one another, but they're also identical to the original parent cell. So here we have daughter cells which are identical, but they're also identical in genetic makeup to the parent cell. But what happens when things go wrong? Something could happen in the copying process. You know how it goes with a photostat machine. Things can go wrong. If you look at uncontrolled mitosis, describe in the question for 4 marks what can go wrong. This is where cancer comes from. Cancers start when changes occur in the genes that control this whole process of cell division. And any change in a gene, please remember this word, is called a mitosis, it's called a mutation. Now, instead of one cell forming 2 identical cells, the cells that are forming are not identical, and now they form a mass of cells, and they form what we call a growth or a tumor. These cells are not normal, but they can be treated. That's the good news. What kind of treatment is there for cancer? Cancer is curable. Please remember this. Describe 3 forms of treatment that have been proven effective. There's radiation therapy, that is high energy of radiation that destroy cancer cells. There's chemotherapy, and then there is surgery. Chemotherapy has to do with chemicals, and surgery has to do with surgically removing the tumor. So if you look at what we've done today, you notice that we have done the different phases of mitosis, and then there's certain terminology. Let's look at these terminology again. Remember, we spoke about the phases, the 4 phases of mitosis, but what about centrioles? Can you remember those tools that form the spindle fiber? Then we spoke about how the cytoplasm divide for cytokinesis, and we spoke about uncontrolled mitosis. So just to recap on what we've done today, let's just look at the bigger picture. We spoke about the different organelles inside the cell. Then we spoke about the government of the cell controlling everything, the nucleus. We spoke about cell division, which is called mitosis, and that leads to growth. And then we spoke about how cancer can be treated. So we've come to the end of this lesson. I trust you've learned a lot, and I can't wait to see you again. Goodbye.